Hello and welcome everyone. My name is Dane Menke. I am the Digital Marketing Manager here at Regenesis and Land Science, and I'll be your host for today's event. Before we get started, I have just a few administrative items to cover. Since we're trying to keep this under an hour, today's presentation will be conducted with the audience audio settings on mute. This will minimize unwanted background noise from the large number of participants joining us today. If the webinar or audio quality degrades, please disconnect and repeat the original login steps to rejoin the webcast. If you have a question, we encourage you to ask it using the question feature located on the webinar panel. We'll collect your questions and do our best to answer them at the end of the presentation. If we don't address your question within the time permitting, we'll make an effort to follow up with you after the webinar. We are recording this webinar, and a link to the recording will be emailed to you once it is available. In order to continue to sponsor events that are of value and worthy of your time, we will be sending out a brief survey following the webinar to get your feedback. Today's presentation will focus on selecting appropriate molecular biological tools to assess remediation solutions and monitor performance. With that, I'd like to introduce our presenter for today. We are pleased to have with us Ms. Dora Taggart, President of Microbial Insights. Ms. Taggart received a biomedical engineering degree from Vanderbilt University and has focused on the optimization and implementation of molecular tools for environmental remediation, microbiologically influenced corrosion, and microbial source tracking. Since joining Microbial Insights in 2001, she has developed and commercialized over 60 different nucleic acid-based analyses. Under her direction, Microbial Insights has become a worldwide provider of molecular tools for leading consulting firms, government agencies, and academia. All right, that concludes our introduction. So now I'll hand things over to Dora to get us started.
All right, thank you very much, Dane, and thank you, Tricia, for helping to set up this webinar and the invitation to speak today. Microbial Insights has worked with Regenesis and their projects for almost 20 years, and through this long history, we've had a really great relationship and learned a lot about the best tools that we can use to help evaluate and optimize performance of the great products that's offered by Regenesis. So during this presentation today, what I hope to do is cover some basic information about selecting that appropriate tool to use at your site. And then also for you more advanced users, I'm hoping to include a few tidbits about some upcoming new information that we have and new projects that we're doing that I think will be very interesting to you and provide some valuable information. So I'm not going to spend a lot of time on this slide because I think most of us today understand the importance of that third line of evidence that microbial data can provide. This is a direct way that we can examine our microbial populations without making assumptions like we have to from our contaminant and geochemistry alone. And I'm going to show you that in some of the case studies today. Also with these techniques, there's no need to grow and manipulate samples in the lab. We know that we just don't do that very well because less than 1% of bacteria can actually be cultured in the lab. So we can use these tools to directly assess our microbial populations and really know what's happening in situ. So when these new tools come out, we're constantly evolving and expanding on our capabilities as we learn new things from research. We tend to get really excited because we've heard about this new tool and we may want to apply it across the board. Well, what I want to encourage you to do today is to start by thinking about the question that you have for your site. Let that question that you have dictate the tool that you'll choose, and that's what I hope to show you through this webinar today. That way you can help to maximize the data that you receive so it makes these tools very cost-effective. We're not doing science projects here. We're using these as cost-effective tools so that we can make better decisions at our sites. So let's talk about the first technology. Most all of you on the call, I bet, would probably say that you've heard about this because it's been used for the past few decades. qPCR is a technology that's been well vetted. We've had a lot of government funding. Different agencies have shown us that this is the most precise way to accurately quantify specific microbial populations that we're interested in. I'm not going to go into the details of it. I'm happy to answer your questions. But with this technology, what we're going to do is either look at taxonomic identification and be able to quantify a specific organism that we're interested in, such as Dehalococcoides. We do this commonly. Or we're going to look at functional genes that we know that are important for specific activities at our site, such as anaerobic benzene degradation. So with qPCR, we're going to extract the DNA directly from our samples. We're not going to culture it. We're just going to do a direct extraction from our groundwater, soil, or our biotrap. And then we're going to run individual assays so that we can target all these genes and organisms that we're interested in. But because some sites and processes can be very complex, there can be mixtures of contaminants, or some contaminants have a lot of degradation pathways, we also have a tool called the QuantArray, which you've heard about over the last couple of years, that's essentially qPCR on steroids and allows us to target many pathways and organisms all in a single analysis. Just like qPCR, because it is essentially qPCR happening, the DNA is extracted, but rather than running those individual assays in different tubes like I just showed you, the sample is going to be applied to a square on a slide, and you can see the zoom out picture here. That square is actually comprised of different through holes, and the qPCR reactions happen in each of those holes. So with these, we get to analyze many different functional genes and taxonomic targets that we're interested in, all in a single analysis. And the benefit is, unlike different array technologies where we've got interactions that can be occurring, because these are happening inside the hole, we're getting no cross reactions. And it's the exact same as if we did them in the tubes, just on a smaller volume. Now, there are different QuantArrays that are available depending on the contaminant that we're interested in. For instance, the petroleum array looks at aerobic genes for BTEX, MTBE, and PAHs, because these are common co-contaminants that we see at sites. We can also look at anaerobic markers for these same contaminants. So in one analysis, you're looking at both your aerobic and anaerobic pathways all at the same time. The QuantArray-Chlor is another array that's available to us, and it lets us quantify reductive dechlorination. And what I want to point out here, we're talking about reductive dechlorination of a number of different chlorinated compounds, not just chlorinated ethenes, but you can see the big list of compounds that we have here, from chlorinated ethanes, chlorinated methanes, and propanes that are available that we can see with this array. We also get to look at organisms that may be competing for the available hydrogen that we add into our samples with these electron donors. So I'm going to show you that in a case study today, why it's really important that we look at these groups as well. And then also on the chlorinated array, we're not going to look at just reductive dechlorination, but we're also going to look at a number of genes that are involved in cometabolic degradation of the compounds. And we know some about cometabolism, but I want to linger here for just a moment, and for those of you that are more advanced, because I think this is some really exciting information that's starting to come out. I think cometabolism may be a much more important part of our sites than we give it credit for. So Dr. John Wilson and Todd Wiedemeyer are currently funded with ESTCP to look at this and to try to understand a little bit better. So this is a presentation that we're going to be doing at the Battelle Conference in Miami this year. But I want to give you guys a little sneak peek of this data. So especially someone that's more advanced, I think this could be very useful for you and your sites. So what we've graphed here are the data from sites where we have TCE degradation rate constants, and we've compared a functional gene that's associated with cometabolism, one of the many that I just showed listed. And I'm just talking about just this one gene today, phenol hydroxylase, or PHE, as we've shown here. So if we focus on the data where there are significant amounts of degradation, those are our red dots on the graph. I've drawn this line here so you can see it a little better. So when TCE degradation is below this line, we see that PHE is low or non-detect. When there's some degradation happening, we see PHE concentrations are higher, around 10 to the 3 cells per mil mark. And then with faster degradation, we see higher concentrations of PHE that are being detected at around 10 to the 4 cells per mil. Now, in looking at those co-metabolic genes, in the past, this isn't one that I would have pulled out and expected to correlate as well as what we've seen. And they looked at quite a few different sites and a number of samples at some military bases. And we're going to add more data to this. And so I hope if this is of interest to you, you'll come join Dr. Wilson's session there at Battelle and kind of get more information on what's going on with this and what we're seeing, because I think it's really exciting. We're starting to grow and learn so much more. Okay, so let's move on from our sneak peek, and let me give you a case study of how we can use qPCR and QuantArray at our sites. This is a typical chemical facility. It has a mixture of chlorinated compounds, ethenes, ethanes, and propanes. And as I mentioned in the beginning of my talk, how I really want you to think about this, start with the question you want to answer. So in this case, we want to know, is it likely that we're going to get complete reductive dechlorination? Do I need to add an electron donor? Is that going to be effective enough, or do I need to bio-augment? So as a side note, Microbial Insights has been processing qPCR samples since 2001, and we have a very large database of samples. So when we look at more than 12,000 samples with the DHC and the chemistry, we see what I've plotted here is that when we have greater than 10 to the 4 cells per mil, we find complete degradation to ethene almost 80% of the time. So as a site manager, I can use this data to give me kind of a marker of where I want to hit with my Dehalococcoides concentration to get good odds that I'm going to have complete reductive dechlorination at my site. So having this number, we can start to put into context what our QuantArray data means at a site. So what I'm showing here are different dechlorinating organisms from one well at this site, just to give you a quick example. And this is our baseline sampling data shown here with the blue bars. As we can see, we have low concentrations of our Dehalococcoides, our DHC here, and then we have 2 other dechlorinating organisms that are important for the chlorinated ethanes that are being detected. Now, Microbial Insights has a database where we've collected all of this data from the sites around the world, and if we take this database, we can start to put this into a little more context. What we see here is that we're at the bottom 25th percentile compared to samples around the world. And that DHC is below that 10 to the 4 mark that I just showed you is really important for effective biodegradation. So now let me add to this same graph. These red bars are going to show the post-injection results. And in the beginning, I want you to focus on the right-hand side of this graph with the organisms that do the chlorinated ethanes and propanes. And what we see is post-injection, we've really stimulated concentrations of these organisms, and for this, we're in the top 10% compared to samples around the world. Now let's go over to that left side of the graph and look at our Dehalococcoides concentrations for our chlorinated ethene degradation, and we see that we're just shy of that 10 to the 4 mark, but this is just post our injection of our electron donor into the environment, and so we're close to where we want to be. And we're already at a point where we're almost 70% higher compared to other sites around the world. So now we can look at the rest of our QuantArray data and try to figure out what happened in this first sampling event that may have caused us not to quite reach that 10 to the 4 mark where we want to be. So in this plot, I'm showing you the total bacterial concentration, shown there as our EBAC number, sulfate-reducing bacteria, and methanogens. Post-injection, we see in our red bars there that we have an increase in both these sulfate reducers and the methanogens, and this is something that we commonly see at sites around the world because these organisms can utilize that hydrogen that's produced from our electron donors. But what we find is that once the bloom in these organisms stabilizes, we commonly see dechlorinating populations start to rapidly decrease. From the data that I've seen, there's typically a 3 to 9 month lag depending on the sulfate concentrations at the site. So if I were a site manager for this site, I would be very excited that I almost got that 10 to the 4 mark in my first sampling event, and these increase in sulfate reducers and methanogens would help me to understand a little bit of why there may be some lag before I see even more growth of my Dehalococcoides populations at these sites. And you've probably seen data from qPCR and QuantArray, so you know that it's applicable to both petroleum hydrocarbon sites like I've talked about, as well as that chlorinated case study that I just mentioned. And I want to point out that these tools are really applicable for most all enhanced remediation products that Regenesis offers. So whether you're adding the ORC Advanced or HRC, I think this is a great tool that complements those products. I would recommend using them in the beginning so that you get your baseline concentration, so you can use that to compare like we did in those graphs and kind of optimize your performance. Then you can do that continued monitoring to make sure that you're still getting effective distribution of your electron donor. Also, if you've already started a site, we can use it just for the continued monitoring as well. So if you don't have that baseline, it's not detrimental to your study. It's just a nice component to have. qPCR and QuantArray are also great tools that you can use with chemical products. So whether you're talking about the chemical reducing solution or the persulfox, these are great tools that really complement it, because while we might see that the microbial community is somewhat suppressed during the time that the chemical products are most active and working on our contaminants of concern, what we find is after the community starts to stabilize and the oxygen or the sulfate is more available and bioavailable to our microorganisms, we can get stimulation of those aerobic and anaerobic processes that really give us that treatment train effect to have complete degradation of our contaminants of concern. Another time that we find this to be very useful is in combination with inoculum injections. So if you're going to be adding biodechlor inoculum, qPCR is a great tool that we can assess the Dehalococcoides populations, and we can understand the survivability of that culture in situ to make sure we're doing effective injections. And then finally, this is something I'm really excited about, and I think for those of you that are starting to use this product, you're probably pretty excited about it as well. But with PlumeStop, we've seen some really interesting data, and I think qPCR and QuantArray can be combined with this to really help you document and understand the biodegradation that happens with these applications. So whether you're adding PlumeStop by itself and just looking at the effect that it has on your microbial populations, or if you're combining PlumeStop with your biodegradation products like ORC for your petroleum hydrocarbon sites, or you're doing the 3DME and the HRC, these are, this is a great tool that we can use with the PlumeStop. So because of this, I want to show you a quick example of something that I found really exciting. This is a TCE impacted site, and here I've shown you the contaminant concentrations as a function of time, just before and after the PlumeStop and HRC injections. So we see initially our TCE concentration was around 500 micrograms per liter, and it dropped immediately to non-detect post our injection, so very effective absorption of our compound. But what about biodegradation? This is where combining it with the molecular techniques can really help us understand and improve what's happening at the site. So after injection of the PlumeStop and biostimulation with the HRC, what we see is our Dehalococcoides populations, our blue bars here, increase, and now we're above that 10 to the 4 threshold where we want to be for effective biodegradation. And we see that Dehalococcoides was maintained at this concentration for about 9 months, even after removal of the chlorinated hydrocarbons from the dissolved phase at the site. After this period, we see what I'm plotting here is our total organic carbon as a representative of our electron donor compared to our Dehalococcoides concentrations. And we see that by 15 months, TOC had decreased to less than 10 milligrams per liter. Dehalococcoides populations were then decreasing by about an order of magnitude. And I didn't show it here, but the vinyl chloride reductase genes found in Dehalococcoides also dropped from around 10 to the 3 cells per mil down to 10 to the 1 to 10 to the 2 cells per mil. So what I did here was rescale this y-axis so we could take a closer look at our daughter product concentrations, because I think this is really interesting. Once electron donor was completely consumed, unfortunately we don't have Dehalococcoides during this period, but I would assume that DHC concentrations probably continued to drop once there was an electron donor that was available. And that makes sense because we make conditions less favorable. We saw ferrous iron decrease, sulfate increase, so a return to a little less anaerobic conditions with consumption of our donor. And once Dehalococcoides decreased, you can see that our vinyl chloride concentrations come up just a little bit. So we're getting very low vinyl chloride that's being formed at this point. Now this can be helpful in providing evidence that reductive dechlorination is happening from our TCE. It wasn't just adsorption. We're starting to see daughter product formation. So the takeaway from this is PlumeStop combined with HRC really resulted in some effective absorption and biodegradation to happen. Dehalococcoides is an obligate halo-respiring microorganism, and there's no way that our populations would have changed at would have maintained at such a high concentration of 10 to the 4 cells for 9 months if they weren't actively doing some type of biodegradation. And when we see that tiny bit of vinyl chloride coming up at the end, that helps to provide some evidence that sequential reductive dechlorination was happening. So it's important to remember that vinyl chloride was only detected once DHC decreased. So if additional electron donor was supplied or a second application happened, even those low vinyl chloride concentrations would likely have been degraded, which leads me to my next point. Microbial monitoring with qPCR is very important with this product to help us understand biodegradation, because based on these results, we may not see the typical daughter products we would use as a line of evidence. So I think if we can combine those this tool with the PlumeStop technology, it's a very good way for us to understand and prove biodegradation is happening at our sites. And what we find in this is a little tidbit of side information for you is we've looked at collection of soil, of groundwater, and what we find the best results on the microbial side is once you do your PlumeStop injections, if you collect groundwater in your monitoring well, you're seeing some turbidity from that PlumeStop, that's a really good place for us to collect our samples, and we can start to see where the organisms have likely colonized around that area of the PlumeStop and started to do the biodegradation as part of the process. So again, you can contact us if you want more information about that, but that's kind of some of the a tidbit of information that we've gained from the last year or two working with Regenesis on this product. Okay, so let's move on to the next tool that I want to talk about briefly today. It's called Next Generation Sequencing. But because of time, I can't go into the great depth about Next Generation Sequencing, but we do have a webinar that's on the resource section of our website that you can use to get more information about the tool. I'm going to give you some brief overview here. Typically, when people refer to Next Generation Sequencing, what we're talking about is community profiling using amplicon sequencing and looking at the 16S gene to give us taxonomic information so we understand who is there. So we're going to want to choose this tool when our question at the site is, how is our community shifting? Say we've added HRC or we've added 3DME at our site. What total community change are we seeing so that we can see, do we have a shift to more anaerobic organisms? Are we getting good distribution of our electron donor? That's a great use for this tool. Community profiling and comparison is the best way to use this type of data from our experience. So let me show you a little bit about what you're going to get with this type of analysis. We're going to get a chart similar to what I've shown here. It lists each sample that was analyzed, and the second column shows us the number of reads that were sequenced in our sample. The third is going to show us the % of reads that we were able to classify. And in that fourth column, we get some information on diversity, so we can start to see which samples across our site have more diverse microorganisms or which may have been impacted by our contaminants, so we had a change in the diversity of our microorganisms. You're also, for each sample, going to get a bar graph like I'm showing here. And this tells you how much of these sequences we were able to classify. Now, typically, when I'm working on environmental samples, I'm going to be looking at the genus level identification. That's really what we tend to find most useful for bioremediation purposes. Also, with each sample, you're going to get a table like I've shown here and a pie chart showing the different genera that were detected in the sample. So the genera of the sample is going to be our variable that tells us the similarities and differences in our samples and give us a lot more depth of information. So what we're going to do is look at this table, look at what genera of organism was identified, and then the descriptions in this table are going to give us generalizations about this particular genus. Now, it may not tell us what every species in that genus can do, but it's going to tell us what some of the microorganisms in this genus can do for us. The thing I want to point out here that I think is really critical is these are relative abundances. Do not try to use this quantitatively. We really need to use other techniques like qPCR to get quantitative numbers of these organisms. But this can give us relative abundances that we can use for comparison. So once we have all these genus identifications for our samples, it can be a lot of different genera. We can have 50 to 100 different genera detected in each of our samples. It's a lot to look at. So with that, we want to move forward to using some type of statistical analysis to help us compare the microbial populations at our site. So what I'm showing here is some analysis done that I like to do on these samples called hierarchical clustering. And what we can see here is which samples are more similar to each other and which are more different. So as you can see here, T3, T4, and T5 are more similar to each other than T1 and T2. So then we can dive into the data a little deeper and find out what's the difference in these samples. And in this case, it was baseline versus post-treatment. So we can use that to help us figure out the grouping. Maybe it's background samples versus plume or down gradient locations that we start to see the clustering happen, but it gives us a lot of information about our site. Then we can take this a step further and use additional statistical analysis such as a principal component analysis or a PCA biplot like I've shown here that can help us understand which of the genera of microorganisms cause the samples to group together. This can help us understand how the treatment affected the populations and help us understand the potential effect on our contaminants. So for instance, if you look at this group in this quadrant, Dechlorination is one of the organisms that caused these to group together, Geobacter over here, and Albinoferax down here. So then we can start to do more investigation into each of these microbial organisms to really understand what's happening at our site. So I want to encourage you that NGS is a great tool that we can use for community profiling so we can compare our samples. It's also great for emerging contaminants where we don't know the pathways, so we can't target specific microorganisms or pathways of interest. So we want to apply it when we want to see a total community shift. As I mentioned before, if we're applying 3DME or HRC, how did our community shift pre and post-treatment? And then we can combine this with our qPCR data to be able to make some really strong conclusions about what's happening at the site. Again, NGS isn't going to be quantitative, and we may miss some of the low abundance microorganisms. So it's a tool we want to use for community profiling. The next part of the talk that I want to discuss are two of my favorite tools, stable isotope probing and compound-specific isotope analysis. Again, I'm trying to go through a lot of tools in a short period of time, so I can't get into specifics, but on the website there's webinars that can give you a lot more depth about these tools that are available. So let's start with compound-specific isotope analysis. This is a technology that's been around for several years that I think is very valuable for us in site management. Before I get started on the technique, I want to point out that there's an excellent guidance document from the EPA that talks about CSIA. So if you're going to be using this at your site, I would recommend that you download this document because it can give you some information on the different isotopes, how to do 2D versus 3D CSIA analysis, and really give you some direction on site management. And before I dive into looking at some more information about this analysis, I want to talk about our units. This is going to be the same whether we're talking about CSIA or stable isotope probing. It's called a delta value is what we're going to get, or a unit of per mil. So it's essentially looking at the ratio of our carbon-13 to the carbon-12 in our sample compared to that same ratio in an internationally recognized standard, PD Belemnite. So we've got a way to standardize that value so the delta value is comparable no matter where we're collecting the samples from. The way that CSIA works is because in nature, our 12 carbon chemical bonds are slightly weaker, and they're going to react more quickly than a 13 carbon bond. So as a compound biodegrades, it's going to become more enriched in the 13 carbon. Due to this, when our daughter products are formed, they're initially going to be very depleted in 13 carbon. So I really like this slide. I borrowed this from Professor Mike Hyman at North Carolina State University because I think it does a great job of illustrating this concept, and I'm a big M&M fan. This is a 13C chocolate fractionation process. In this illustration, each M&M represents a molecule of a contaminant. If the contaminant is primarily carbon-12, it's blue, carbon-13 is yellow. So if we go from left to right in our beakers, what we see is the contaminant, our M&Ms in this case, are being consumed. So if we imagine there's a preference for the blue M&Ms, our carbon-12 ones, and as we look left to right, we notice that the overall number of M&Ms is decreasing, but the proportion of yellow to blue is increasing. That's analogous to what's happening in our naturally occurring contaminant. The 13 carbon content in the undegraded pool is increasing over time, so we can use this to understand degradation processes. As I mentioned, CSIA has been used very successfully over the last 5 to 7 years to really provide a line of conclusive evidence of biodegradation. It's applicable to a wide variety of compounds from chlorinated ethenes to 1,4-dioxane. Anything that the organisms use as an electron acceptor, I really like to choose CSIA as my tool for this. So especially for chlorinated compounds, this and qPCR are a tool that I would commonly select in site management. It gives us a way that we can really estimate how much our parent compound is biodegrading. We can also use this to help us understand abiotic mechanisms and degradation in our population. And then finally, it's one of the few tools that we can use in environmental forensics where we can get source identification. If we have multiple sources we need to delineate, this is a great tool to be able to do that. When we link it to the Regenesis products that are offered, we can use it with pretty much any of the products that are being used, like the 3DME, HRC, the chemical products, ORC Advanced. So we can use this as a complementary tool to help us understand the biodegradation effects from these compounds. Again, I want to give you just a little side bit of information from a new case that we're working on. This is another presentation that's been submitted to Battelle. It's where 1,4-dioxane, where CSIA was used to help us understand if biodegradation is happening across the plume. This is a sneak peek of the data from the hydrogen and carbon analysis that we have so far on this site, and this is our baseline data. So what you see is the concentrations of 1,4-dioxane seem to go down as we go down our plume. So we wanted to understand if this is biodegradation that's occurring. So we did delta 13C analysis and then the hydrogen analysis as well. And what we found is there's not really any indication from the CSIA that biodegradation is occurring. It seems like there's another process like dilution or something that's happening at this site. So what the client decided to do from this is they're going to move forward with a bioaugmentation of CB1190. Then we can use qPCR to help us confirm the survival of the CB1190 after it's added. And not only did it survive, we can use CSIA to help hopefully show that it starts to cause biodegradation of the 1,4-dioxane in this plume. So again, this is something that's going to be presented more in full case study with hopefully the results of this next round after the bioaugmentation at that Battelle conference in Miami. Okay, and another tool that we can use that's very complementary to CSIA is called stable isotope probing. This is a tool that we can use to conclusively confirm biodegradation in situ under actual site conditions. So we know that in nature, 99% of our carbon is a 12 carbon and only 1% is 13 carbon. So if we flip that ratio and synthesize a compound that is 99% 13 carbon, it's still going to behave just like our original molecule. But then we can use looking for that 13 carbon in the environment to help us understand the fate of that contaminant. The way that it works is we're going to take our specially synthesized compound, and we want it to be something that the organisms use as a carbon and energy source because I want to track that into degradation products. So for instance, in this case, we're going to do a 13C labeled benzene. We're going to load it onto our biotrap samplers. Biotraps are a tool that's unique to Microbial Insights, and we'll talk about those in just a moment, but the biotraps are loaded with a known concentration of benzene, for example. Then they're put in a monitoring well for 30 to 60 days. Following deployment, the biotraps are retrieved and returned to the lab for analysis. So before I move into showing you a case study of this, I want to go over what a biotrap is. This is a passive sampling tool that was designed by the University of Tulsa and DuPont. We've used this for the last decade at sites around the world, and it's a way that we can collect microorganisms so we can understand biodegradation potential. Now the key to the biotrap is inside that slotted PVC that you see. We have bioset beads, which are about the size of a BB. These are composed of 75% powder-activated carbon and 25% Nomex. The pack is going to give us that absorptive capability that we need to add our compound or to absorb natural compounds. The Nomex is the same material from a fire retardant suit, so it's going to allow us to fire these beads at very high temperatures so we can make sure they're sterile. So the organisms that colonize these beads in the monitoring well came from our well there. So if we have these compounds sorbed to the bead, put them in a monitoring well, we can see if these organisms utilized our compound of interest. And there's a lot of surface area inside each of these beads, so it's a great place for the organisms, the indigenous organisms in that monitoring well, to colonize, form biofilms, and start to utilize that compound of interest. So what we're going to do is, once these are returned to the lab post-deployment, we're going to look for our carbon-13 in 3 different areas. We're going to look, we know how much was put on the biotrap originally and how much is remaining. More importantly than what was lost is where did that loss go? So we're going to start to look to see if the organisms mineralize that carbon, looking at the dissolved inorganic carbon or the carbon dioxide, and see if we have a higher 13C ratio in our dissolved inorganic carbon. And we're also going to look in the phospholipid fatty acids, because this is how we look at the cell membrane of our microorganisms. So if they utilize as a carbon and energy source our 13 carbon from our compound, they're going to incorporate that into new biomass that they build for cell growth, and then we can document that with PLFA analysis, looking at those cell membranes, and prove that the organisms utilized this compound. So let me give you a quick example of where we use this again at a site. This is a typical MGP site. It's been in operation for a long time. They'd removed a lot of the source area compound. Mainly what they were left with of concern were benzene and naphthalene. And from looking at the contaminant concentrations and the geochemistry doing our Mann-Kendall trend analysis, we saw that it looked like biodegradation was happening. Things were pretty controlled at the site. But the regulatory agency really wanted further proof of biodegradation. They wanted that third line of evidence from the microbiology. So again, what we started with is, what's the question we have at the site? The first question is biodegradation occurring? And then if we prove it's occurring, how do we have some type of tool that we can use to continue to monitor our contaminant concentration so we can be sure that these organisms are maintaining over a long period of time? So we're going to use stable isotope probing and the QuantArray. So I want to show you just a little bit of the data from the site. And I'm going to focus just on the naphthalene here because of time, and this is a pretty large case study. So there were 3 monitoring wells that I'm going to be talking about today. I want to talk about this 7C well primarily for our stable isotope probing because this is one of our source area locations. There's a down gradient location that we're going to look at, and then a background well that we're also going to look at with the QuantArray data. And I'm going to hopefully show you how these are very complementary tools that we can use. Okay, so here's the stable isotope probing data. Now this is our delta values from our dissolved inorganic carbon. So in nature, if we did not have a 13C compound that was added, we would expect our carbon dioxide to have a negative delta value around a -25. But in our source area well, what we see when we analyze our biotrap samplers is we're finding carbon dioxide with a positive delta value almost at 250. There's no way that this could happen unless the organisms were mineralizing that 13-carbon naphthalene and releasing it into the carbon dioxide. So this is proof positive in this line alone that the organisms are utilizing our compound of interest. But we also look at it into biomass because not only can they blow it off in respiration products, but they can also incorporate it to build new biomass to do more degradation. And we see the same thing when we look at our phospholipid fatty acids. In background, if we had no compound, we would expect a negative delta value. And look at our delta value here. We're almost at around 850 that is incorporated. So proof positive, if the naphthalene is bioavailable, the organisms are utilizing that naphthalene that we put in the site. So now let's look at the QuantArray data. Again, I'm just pulling the genes that are important for naphthalene due to time today. I'm just going to talk about this. So we've got our 3 wells here that are showing plotted. These are our aerobic genes for PAH degradation and our anaerobic ones. And what I want to point out is our MMW17D well here. Look at this. It's non-detect for every one of these genes. And I have to say that would not commonly be what we would see. Typically, there may be some of these organisms that have the genetic potential, but they're not active at the site. But in this case, our background well shows us there's nothing there. So unless the naphthalene has been there at some point, we're not seeing these organisms naturally at this location. But in our source area well and our down gradient location, we're getting some aerobic PAH potential and quite a bit of anaerobic PAH potential, especially in that down gradient location. And this gene here is anaerobic naphthalene carboxylase. It's a gene that was recently identified for anaerobic naphthalene degradation. And look how high we are, especially in that down gradient location. So when I was presenting for the state agency in this case, I was able to go to our database, look at this gene concentration and say, you know, we're higher than what we've seen at 96% of sites around the world. So we could make some very strong conclusions using that stable isotope probing data and that QuantArray data about what was happening at this site. So to conclude what we saw here, the chemical lines of evidence were a green light, looked like things were pretty stable. Then we used this microbial data to help give us that third line of evidence to prove that biodegradation was happening at the site. And on the QuantArray, we had both aerobic and anaerobic potential that we could use for monitoring. And we've seen that SIP can produce this conclusive data from a lot of different compounds and pathways. The technique has been used on a wide variety of compounds. For any BTEC sites or PAH sites, even 1,4-dioxane, I think it's a very valuable tool. We've used it at several chlorobenzene sites. And then for emerging contaminants like dioxane and sulfolane, it's a really good tool that can help us prove biodegradation in a site. The other thing that we can do with the stable isotope probing is it can help us prove how effective a remediation product is going to be to enhance our microbial population. So what I'm showing you here is just a really quick example, and this is going to lead into in situ microcosms that I'm talking about. But what we can see here is our 13C incorporation into biomass looking at our phospholipid fatty acids. We can see under natural conditions, we had incorporation. There's biodegradation that's happening. But when we add our ORC advanced, look at the difference in the amount of incorporation into biomass. So this is a great tool that we can use to help us understand the efficacy of using different products. So that brings me where I'm going to switch gears just a little bit, and I want to talk about, as my final tool today, in situ microcosms. These microcosm units are an adaptation to our biotrap samplers, and they provide an excellent way that we can evaluate the performance of different electron donors and acceptors in the field. And we do this commonly with a lot of the Regenesis products. These microcosms are all custom made based on the question that we're trying to answer at the site. So if we want to compare different treatment options, then we can do this in situ. It's kind of similar to what we would do in some bench scale studies. For instance, if we have a petroleum hydrocarbon plume, we're trying to decide if we want to add an ORC or if we want to add a sulfate. We can look and add a control trap, one with ORC and one with a sulfate amendment, and then help us understand aerobic versus anaerobic pathways and deploy these in the monitoring well at the site. Typically, for petroleum sites, we're going to leave them 30 to 60 days. For chlorinated sites, we may leave them 60 to 90 days. The way that it works is the traps are attached in assembly of units, and each of the units has baffles on each end that can help us segment off a portion of the monitoring well. Now, these are just slotted PVCs that have the baffles, and to be honest, the baffles work much better to help minimize crosstalk between the units than I ever thought. Inside each of these units, what we're going to see is typically at the top and bottom of the unit, sponges that we can use to add our liquid electron donors or acceptors. We can also have porous bags that we use for solid amendments. Then we're going to have a passive diffusion bag that we use to look at our VOCs and our dissolved gases, a biotrap to look at our microbial populations, and finally a vial that we can use for our geochemistry. And so by combining data from these different multiple lines of evidence, we can start to get a really much better understanding of what's working at our site so we can make very cost-effective site management decisions. So let me give you a quick example before I wrap up today. This is a typical chlorinated site, TCE contaminant, DCEs detected, not a lot of vinyl chloride or ethene that's being seen at the site. So their site managers considering biostimulation or bioaugmentation. So as I mentioned, we want to start with what our questions are. So from the 3 units, the control unit can tell us if Dehalococcoides is present under natural conditions. The biostim will show us what happens if electron donor is added. Are we stimulating those halo or spiring bacteria, or if bioaugmentation is going to be needed? And then if we bioaugment, we can check to see if the culture is going to survive in situ. So what we can do is take our biotrap, load it with the biodechlor inoculum, and put it in this bioaugmentation unit. From the chemical side, there's a number of questions that we can ask. We can look at what the contaminant looks like in our units under natural conditions, and then how that changes with our electron donor addition. Are we getting full reductive dechlorination? And then how much does it enhance when we have the bioaugmentation? So here's quickly the microbial results from this. Our Dehalococcoides populations and vinyl chloride reductase genes are pretty low in our unit. We're around 10 to the 1, so there's some there naturally. We see an order of magnitude increase even in a short deployment time of around 60 days in our biostimulation unit. And then we see in our bioaugmentation unit that the Dehalococcoides concentrations are similar to what we had pre-deployment. So from our microbial lines of evidence, we see that we have a low indigenous population, and that when we add an electron donor, we're getting a noteworthy increase in our bacteria. So do we need bioaugmentation? Probably not, depends on the time frame. But if we do need to do bioaugmentation, we can see that the culture will survive. What does our second line of evidence show us? This is the VOC results compared between units. We can see in the biostim unit that we have our TCE is now pretty much gone. Primarily we got DCE that's present at the site, and we're starting to see some vinyl chloride and ethene formed. And then we have a little bit more vinyl chloride and ethene being generated in our bioaugmentation unit. So from our chemical lines, we see that under existing conditions, we look similar to what the site managers were seeing, mostly TCE and DCE. With donor addition, with our HRC in there, we're getting enhanced DCE production around 90%, so we can get complete reductive dechlorination from those bioaugmentation, or from those daughter products we're seeing, the vinyl chloride and ethene. And so we can see too that if we do bioaugmentation, we are getting some enhancement, but you know, not a huge amount of enhancement over the biostimulation. So from the site management decision, then the client can make the decision based on time frame and other factors of what they want to do moving forward. And then in this case, they decided to move forward with biostimulation. So hopefully, as you can see, this is a really effective tool that we can use to understand in situ how a compound and how the microbial community are going to react to particular amendments. And by using these multiple lines of evidence, we don't miss something. We get a really good view of the changes in the chemistry, geochemistry, and microbial community. And it's a very economical way for us to be able to evaluate products and pathways in the field, not on a lab bench, but in the field, prior to moving to a full field scale application. So as I close today, I know some of you on the webinar may not be as familiar with us. Microbial Insights was founded in 1992. So next year is going to be our 25th year in business. We were a technology transfer from research that Dr. David White did at the University of Tennessee. And everywhere I travel around the world, it seems like people were familiar with Dr. White. He was a really innovative researcher that was so far ahead of his time, looking at microbial community composition using tools that were commonly used in the medical industry in the environmental world. So one of the first, I think, to bring all this to us in environmental remediation. And throughout this history, we've worked with companies like Regenesis, helping understand how our products, how do we effectively enhance degradation in the field and be very cost effective in our management. So now these tools really aren't science experiments. They really are ways that we're learning how to better manage our sites. So before I close, I just want to encourage you that we have a lot of experience here at Microbial Insights, and we've looked at tens of thousands of sites around the world. We do strive to have accuracy and precision in our data. We've been validated in many government studies, different laboratory splits with samples, and we always come out the best. We have good QA, QC. We care about the data, because if you don't get effective data that you can count on that's good quality, then it's a waste of money. We want you to feel like you can get a good value for your money. Also, we want to be innovative, continuing to bring you a better suite of analysis. And as these new products, like the great product PlumeStop that come out from Regenesis come out, we want to have tools that can help complement these products. So we're continuing to evolve and develop new technologies. Also, I encourage you to use the database, because there's more microbial data there than anywhere else in the world. So I think you can get good context to help you make some really strong conclusions at your site. And then finally, just like Regenesis, Microbial Insights strives to have excellent customer service. We want to be a resource to you. If you have questions, even if it's on a technology we don't offer, we'll do our best to answer those questions for you, or to get those answers if we don't know them. So I'd encourage you to go to our website. There's a lot of information on there about these tools, and we have project managers here that can help you, so feel free to give us a call at any point. So at that time, I guess, Dane, I'm finished, and I'll be happy to answer questions that people may have.
All right, thank you, Dora. That concludes the formal section of the presentation, and at this point, we'd like to shift into the question and answer portion of the webcast. Before we do this, just a few reminders. First, you will receive a follow-up email with a brief survey. We do appreciate your feedback, so please take a minute to let us know how we did. You will also receive a link to the webinar recording as soon as it is available. All right, so let's circle back to the questions. First question, Dora, is what analyses would you recommend for evaluating transition from active treatment to M&A and long-term management?
That's a great question. So it depends on the contaminant that we're talking about. If it's something that's used as a carbon and energy source, I like to start with the stable isotope probing. Use that as a tool to prove that biodegradation is still occurring under those current conditions. And maybe even complement that with some of the qPCR for those targets, so you can have a long-term monitoring tool. Then you can do quarterly, semi-annual, or annual monitoring at that point to see that those microbial communities are staying. If it's something that's used as an electron acceptor, I would recommend a combination of CSIA and the qPCR, so we can do that same type of thing.
Okay. All right, next question is, when do you recommend CSIA versus SIP?
Okay, that's a good follow-up to what I just asked or answered. So the CSIA is when something is used as an electron acceptor, like chlorinated solvents. That's probably the best way to prove biodegradation with chlorinated compounds. If it's something that's used as a carbon and energy source, like our petroleum hydrocarbons, I like the stable isotope probing for that.
Okay, all right, sounds good. So here's another question here. Can Microbial Insights use monitoring well groundwater grab samples instead of biotraps for qPCR and QuantArray analyses?
Yes, we can. And that's pretty much, I would say, probably 70 to 80% of the samples that we get are groundwater samples from monitoring wells, and they can be collected either as a grab sample, or when you're doing pump filtration, there's different filters that we can send you where you can collect those in field to kind of minimize the cost in shipping.
Okay, all right, thank you. So let's see here. There's another question here is to review, can qPCR be used to identify and quantify genus, or in parentheses it says taxonomy, of bacteria present, as well as identify and quantify specific function of genes?
Yes, it can do both, and it depends on what we choose as our target. So we can choose to look at an organism taxonomically, like a Dehalococcoides, or we can choose an assay that targets a functional gene, like the vinyl chloride reductase gene for Dehalococcoides. So it can do both of those. We just have to, it's not like NGS where you're getting all the taxonomy. We have to pick which organism we're trying to quantify and use an assay that's specific to that organism or gene.
Okay, thank you. All right, so next question here is, have you ever applied this to an in situ thermal treatment to see the effects on the microbial population adjacent and down gradient to the treatment area?
That's a great question, and that's something that we're actually talking with some of the thermal companies about right now, because I don't think we've done it enough, but we have done it at a couple of sites, and what we see is around that area where the temperature isn't quite as high. In this one particular site I'm thinking about, it was a chlorinated solvent site, and we saw actually enhanced degradation in a halo effect where we're outside that heated area. Microbial populations were stimulated. And in that case, we think maybe there was some cometabolic degradation that was happening in that area for those chlorinated compounds.
Okay, all right, great. So looks like one more question here, and that is, I've heard a lot about different arrays that are available. How are these similar or different from the QuantArray?
Okay, that's a good question. There's a lot of differences in there. So the typical array platform is going to be only taxonomic. I only know of one functional array that's out there, and that's Zhou Zhao's functional array. All the other arrays that are out there are taxonomic identification, and they are not quantitative. It's not an effective way for us to quantify the microbial populations like we can with qPCR. So those are the two of the big differences. The other thing is with array platforms you have to be careful because everything can interact on those array slides, whereas with the QuantArray, everything's happening in those individual holes, so we don't have cross-reaction and get as many false negatives and problems as we can see with other array technologies. And we've tested a lot of different arrays over the years and looked at bringing them in-house, and we ended up choosing the QuantArray technology because we felt it was a far superior technology for site management.
Okay, all right, and actually we do have a couple more minutes left here, and we are starting to get a couple more questions in, so I'll ask this one that came in. What is the approximate cost of C13 testing per sample?
Okay, that's a good question. And it depends on the size of the contaminant. So the smaller the molecule that has to be synthesized for 13C, the more cost effective it is. So for instance, with benzene, you're talking around $1,200 to $1,500 per sample, and that's everything. That's the report, that's all the different analysis, the dissolved inorganic carbon, the lipid analysis, and the interpretation of all that data. Other compounds can be much more expensive. For instance, there was a project where it was a very large compound, and it cost just for the compound itself $30,000 for a gram of the compound, and that can do about 10 biotrap samplers. So it can get very expensive when the molecules are large, but again, it's the only way that we can truly prove biodegradation in the field. So there are times that I guess you can justify the cost if you can bring that level of confidence to the biodegradation.
All right, great. Thanks very much, Dora. That will be the end of our chat questions. If we did not get to your question, we will follow up with you after the webinar. If you need immediate assistance with a remediation solution, please visit regenesis.com to find your local technical representative, and they will be happy to speak with you. If you'd like more information about molecular biological tools from Microbial Insights, you see the contact information there that you can use to get in touch with them. Thanks again to our presenter, Ms. Dora Taggart, and thanks to everyone who could join us. Have a great day.
Thank you.